Good evening, delegates. We warmly welcome you to this 6-day Atal sponsored FDP on the theme of revolutionizing healthcare, the role of artificial intelligence in drug development, clinical trials, and practice. It is a 6-day event in an online mode starting from today, 13th of October, and ending on 18th of October. We will have a number of sessions including about 11 lectures both from academia and private industries. We will have an amalgamation of lectures from experts from academia, research and industry. There will be hands-on session and this is a joint effort by IIIT Allahabad and Molecular Solutions Private Limited. So in this FDP, you will be getting a glimpse of research done in research labs, academia, and how industry perceives it, what is the role of the industry in drug development through AI, and how far they have got success, their achievements, their endeavors, and the purpose behind organizing this Atal FDP is to give information about this emerging field which has a multidisciplinary role and which has a background which is of multidisciplinary nature, and there is a role for almost every aspect of science and engineering into drug development. So I hope in all these 6 days you will get a glimpse of how you can participate in this research area which is of use of application to academia and industry both. So we warmly welcome you and now it's over to Aditi.
Thank you, ma'am. Good evening, everyone, esteemed dignitaries, guests, faculty members, and valued participants. It gives me immense pleasure to welcome you all to the inauguration ceremony of AICT Atal sponsored 6-day faculty development program on revolutionizing healthcare, the role of AI in drug development, clinical trials, and practice, organized by Department of Applied Sciences, Indian Institute of Information Technology, Allahabad, and Molecular Solution Private Limited. We are truly honored to host this academic gathering under the banner of AICTE Atal Academy, bringing together eminent speakers, researchers, and young faculty members from across the country. To mark this auspicious beginning, let us commence with the traditional lamp lighting and Saraswati Vandana. The AICT Atal Academy envisions empowering educators nationwide by providing high quality training in emerging domains, including artificial intelligence, data science, cybersecurity, and healthcare. Through initiatives like this, FDP and Atal strengthen research and leadership capacities, aligning with the national education policy to promote multidisciplinary learning and societal impact. Carrying forward this vision of academic excellence and technological innovation, let us now look at our very own institute, IIIT Allahabad, a premier center that truly reflects the spirit of interdisciplinary growth and learning. Established in 1999 and declared an institute of national importance in 2014, IIIT Allahabad has emerged as a hub of technological innovation and academic excellence. Our scenic 100-acre campus fosters research, creativity, and collaboration across diverse disciplines in information technology and allied sciences. And at the heart of this institute's multidisciplinary growth lies one of its most dynamic divisions, the Department of Applied Sciences, where the convergence of biology, chemistry, and computational intelligence drive innovation. The Department of Applied Sciences plays a vital role in integrating the frontiers of life sciences, physical sciences, and computational technology with postgraduate and research programs in bioinformatics and biomedical engineering. The department is actively engaged in research areas such as drug design, nanotechnology, computational biology, and green chemistry. Our coordinators, Dr. Nidhi Mishra and Dr. Sintu Samantha, have been leading research efforts in these areas, focusing on drug discovery, antimicrobial peptides, and biofilm resistance. Their dedication and vision have been instrumental in conceptualizing and coordinating this FDP, supported by our organizing team and volunteers whose tireless work made this event possible. To further enrich this program, we are joined by leading experts and researchers from premier institutions contributing their experience and knowledge to this FDP. Now, it's my privilege to invite our esteemed director, Professor Mukul S. Rao, to address the gathering. His visionary leadership and unwavering commitment to academic excellence continue to inspire the progress of our institute. We honored by your presence today and eagerly look forward to your words of wisdom and encouragement. We request you, sir, to kindly address the gathering.
Thank you, Aditi. Am I audible?
Yes, sir.
Okay. Good evening, one and all. Warm but virtual greetings for this 6 days Atal FDP on revolutionizing healthcare, the role of AI in drug development, trials, and practices. At the outset, let me congratulate and appreciate my colleagues, Dr. Nidhi Mishra and Dr. Sintu Samanta, along with their team of research scholars and my other faculty colleagues at Department of Applied Sciences, IIIT Allahabad, for conceptualizing this multidisciplinary program, applying to AICT for receiving the approval for this program. And then there are 100 plus participants. I'm really delighted to note that there are 100 plus participants who have joined online this Atal FDP. Guys, friends, ladies and gentlemen, this happens to be a capacity building program, capacity development program in this very happening field of AI for healthcare. Aditi just now very aptly described our institute. IIIT Allahabad is not only an institute of IT, though it was mandated to be developed as a center of excellence in IT, but as IT and allied areas, as she rightly described. So we have very strong Department of Applied Science. Our flagship programs are BTech in IT, BTech in Business Informatics, BTech in Electronics and Communication. We have other MTech 11 programs also. But my Department of Applied Sciences, which is very strong in multidisciplinary research, they also offer two relevant programs to the society. Master's program in Bioinformatics and Master's program in Biomedical Engineering. We have a team of researchers, hardcore researchers, faculty colleagues like Dr. Nidhi and Dr. Sintu in our Department of Applied Sciences, which is a multidisciplinary department. It has faculty colleagues from maths, from physics, chemistry, and also very strong from biology. As again Aditi rightly described, we have all computational intelligence which is embedded with the fundamental sciences of biology, physics, chemistry, and maths. We have computational intelligence. Guys, all of you know that AI now happens to be quite a 70 years old field. In 1955-56, John McCarthy, he coined this term in one of the Dartmouth conferences, artificial intelligence. Human civilizations, we know that we are at the apex of animal kingdom, human beings. We are the most advanced, most progressed brain. And the scientists and engineers and technologists, they have always been tried to probably emulate, mimic this kind of tremendous brain power of human cognitive power, we can say. Okay, the term artificial intelligence was really probably conceived and conceptualized by John McCarthy and then probably Alan Turing, with the concept that can we develop a machine, a mechanical machine is something which can be controlled by human beings, which can mimic human intelligence. Okay, it's a machine intelligence, machine which can think like human beings, and there AI was born. AI again went through various cycles of dips, cycles of peaks, and so on. But guys, understand today is the golden age of AI. AI cannot, no field of human beings can remain isolated from AI. Okay, my generations have witnessed the revolution in terms of internet, in terms of Google, in terms of email, in terms of mobile phones, in terms of radio and television. AI happens to be all pervasive but invasive kind of technology which has come to us today. And AI is certainly embracing and encompassing all walks of human life. How healthcare can be isolated from AI? It cannot be. AI happens to be a tool, it's a technology, it's a multidisciplinary technology which has tremendous applications in all walks of human life. Let it be business, let it be agriculture, let it be education, let it be any weather forecasting, let it be anything, or let it be healthcare also. What has been happening is if you let's say have CT, MRI scans, and then I go to a doctor and want to really diagnose whether there is a brain tumor or a breast cancer for a particular patient, right? And the doctor, probably the cancer specialist, he reads those images and then he decides something based on the image. Okay, but AI today can help us in medically diagnosing very correctly, very accurately, with the highest degree of credibility and performance, whether the breast cancer or brain tumor is present or not. So AI I can use for medical diagnostics. In general, when humans can decide, when humans can estimate, when humans can forecast, when humans can probably comprehend, when humans can create, AI today has all that power, right? AI when it clubbed with neural networks, as late as in probably after machine learning era of 1990s, it came into existence in 2000, let's say, neural networks. Then artificial neural network being deployed in AI, that really paved up a way for immense progress of AI touching all applications of AI. Okay. And therefore, when we have deep learning model, deep neural networks, and so on, the models are evolved from simply probably multi-layer perceptron. Then we had large language model, then long short-term memory model, so on and so forth, transformer model, and so on and so forth. We have many models of AI which can be deployed, which are neural network based, and they can certainly help human beings, assist human beings in, let's say, healthcare for diagnosing the diseases. Application of AI for probably drug development could be new, but remember again, AI happens to learn from data. AI happens to learn from data. Just like humans learn from observations, we learn from childhood, we learn from observations, we learn from samples, we learn from experiences. AI exactly mimics them. So for drug development, using AI probably will certainly need huge amount of vast amount of data. I'm more than sure that in the 6 days of proceedings of this FDP, my colleagues along with the external experts, they certainly enlighten you that how this data driven tool of AI can help us in developing novel drugs. How it can help us. Okay. So also in clinical trials, so also in medical diagnostics, and so on and so forth. Okay. As I told you, that AI happens to be a very powerful tool. And today, the latest development of AI happens to be explainable AI. The decision made by AI, if let's say doctor makes a wrong decision about the disease for a patient and it simply frightens or scares the patient, then we can hold doctor responsible. Is it not? In the same way, if AI wrongly predicts or wrongly diagnoses that a particular patient has a particular disease, then we can hold AI responsible. That's a today's era. You have to see to it that credibility or responsibility of AI has been manifold increased today. So AI will keep on evolving. It will keep on touching or embracing all walks of human life. And it's a very right step and right juncture at which my colleagues have thought of this very deep program. And I should also thank molecular technology authorities that they have joined with us, joined hand with us, and collaboratively we are going to run this program. All the best to all of you for learning. Learning experiences always for the humans, right? Now AI also learns from experiences and data, as I rightly said. I won't don't wonder that in days to come, this Atal FDP on revolutionizing healthcare would be conducted by AI agents, and the days are not far off. We won't need humans, and the fear is there whether humans will be redundant or not. We have to see to it that AI, general intelligence, or AI super intelligence doesn't surpass or doesn't overcome human intelligence. Right? That's a kind of age we have to always remember. I wish you a very happy learning for all the 6 days. I again appreciate and congratulate my colleagues, Dr. Nidhi and Dr. Sintu, for conceptualizing this program. And message to all of you: just keep on learning and see to it that human intelligence always superpowers and outperforms artificial intelligence. Thank you very much.
Thank you very much, sir, for your inspiring and insightful address. Your words have truly set the tone for this faculty development program and will continue to guide us throughout the session ahead. Next, we would like to invite Professor Pramod Kumar, Head of the Department, to address the gathering. Under his dynamic guidance, the department has consistently fostered interdisciplinary research and innovation, serving as a bridge between science, technology, and human welfare. We request you, sir, kindly address the gathering. I request you, sir, to kindly address the gathering.
Sorry to interrupt, Aditi.
Yes,
Just
sir.
now I had a talk over with Dr. Pramod, sir. He's telling we'll be joining in 2 minutes. Okay, so maybe need to wait for 2 more minutes. He'll
Okay,
be
sir.
joining soon.
Yes, sir.
Just I'd like to thank Director sir for your kind timing, and just happy to share that the participants, I think more than 300 participants has registered. Maybe they will join soon. Pramod, I think has joined. Aditi, because Pramod sir was not there, na, so can you please announce one more time?
So he is there.
He is there, but
He is
he was
joined.
not there that time. He
We
just announced
welcome
one
you,
more time.
sir.
Welcome, sir. I'm just asking to.
Yes, sir. Next, I would like to invite Professor Pramod Kumar, Head of the Department, to address the gathering. Under his dynamic guidance, the department has consistently fostered interdisciplinary research and innovation, serving as a bridge between science, technology, and human welfare. I request you, sir, kindly address the gathering.
Uh, okay, thank you very much. First of all, welcome to all, and good evening, everyone, and welcome to this Atal sponsor 6 day FDP program organized in Triple IT Allahabad. So, I am welcoming as a head of department, uh, as a head of department from applied science. So, first and foremost, I would like to welcome our esteemed speakers, faculty members who have taken the time to come to this seminar program and share their knowledge and expertise with young minds. All the young minds will get the opportunity to learn from our experts. I would like to welcome all the participants who have made an effort to attend this FDP program. Your presence here today demonstrates your desire to learn and grow, and I am looking forward to learning and growing with you. This FDP program is chance for us to challenge ourselves and widen our perspectives. Today FDP program is an important event for all of us. as it will provide us with valuable insight and information on the topic that is essential to our education and future careers. So, as the head of department, I would like to tell about little bit about the program happening in Triple IT Allahabad. In our department of applied science, basically we have three STEAM program. One, of course, is related to bioinformatics and biomedical engineering. Second, we are going to start one program in the quantum information technology. And in addition to that, we have our colleagues, they are working in different domains in bioinformatics, biomedical engineering, mathematics, and physics. With this all, once again, I am welcoming all of you to attend this program. In addition to that, I am also I would like to thank both the organizer, uh, Dr. Nidhi Misra and Dr. Sindhu Kumar Samanta for organizing this conference. Thank you. Thank you very much.
Thank you so much, sir, for your thoughtful and encouraging address. Next, we would like to invite Dr. Raghu Rangaswamy, CEO of Molecular Solution Private Limited, collaborator of the FDP, to address the gathering.
So thank you. Good evening, everybody. Good evening to the director, the head of the department, and to all the participants. Thanks a lot for giving us the opportunity to be a partner and a speaker in this event. And I admire IIIT Allahabad for conducting this Atal FTB program on the role of AI/ML and drug discovery and development, which is really a need of the hour now. I'll tell you, and I'm associated with IIIT for last 15 years. And now I'm part of the Molecular Solutions, where we are partnered with more than 15 companies. In fact, things and technologies keep changing. The one thing which is permanent in this world is always change. And now the change is on the AI/ML, which is a game changer, particularly in the drug discovery. Because why I'm telling drug discovery is, drug discovery takes about 15 years to discover a drug molecule. Now with the AI coming in, the time is being reduced to half, nearly probably 5 to 7 years, because in fact, identifying a lead molecule or anything is reduced drastically. The reason is today you can screen, you all might have used only the kind of a docking technologies, but today a new method which has come in, which is called as chemical space docking, where you can start with a billion and go to 10 to the power of 26, that is 100 septillion molecules within a week. And use the generative AI is another technology which has come up, where you can generate billions of molecules as per your target protein in a short time. And now FDA says no more animal experiments. But thanks to AI/ML, which has come in, where all the clinical trial data are accumulated, and today you can generate and you can calculate more than 220 ADME parameters based upon the human data. And you can also, the other beauty thing is in the drug discovery, where all the things is a challenge is you might be having a molecule having a good binding affinity, but it might face some ADME parameters. So it is something okay, very experienced person only can help to optimize. But today the AI/ML helps in okay, how can you optimize a particular property using the computational methods. And you can also check whether a molecule is patented and other things well in advance. And once you have predicted all these things, you can predict the binding affinity of a molecule within about zero point 0.7 to 1 kilocalorie per mole using a technologies like free energy perturbation within 3 hours. Now with the quantum computing, what is coming in, today it is possible to predict within about 5 to 10 minutes. And you can predict the synthetic feasibility of a molecule using AI/ML and rule-based approach. Predict a molecules okay, how can you synthesize a molecule at the lowest cost at reduced number of steps. You can also predict the polymorphic form 100% accurately using AI/ML and quantum mechanics technologies. And even coming to the formulation development, today the AI/ML is playing a very crucial role, because for this API, okay, what is a solvent okay, you can screen about something like 9,000 solvents within about 8 hours. You can predict okay, what is the ingredient which is suitable, okay, what is other compatible things, or what is the stability of this molecule with this combination. You can predict all these things within about 2 to 3 days. So that is the beauty of it. And now with the, like as the director was telling, today we are all in an era of LLMs. So now the LLMs is again changing the scenario. LLMs, AI agents, generative AIs. These are the game changers where, like, let us imagine, like, okay, even you have hundreds of softwares or whatever, hundreds of databases. Just by a command, you can go and search from these databases. You can, like, if you're doing a docking, you can just say, like, okay, this is the molecule database, just dock it with the softwares and give me the results. It automatically does everything. Today, like, we are in a way the AI will replace, like, okay, it's replacing so many things, making things much faster. Today, there are always, like, people say AI is going to replace technologies, people and other things. I would say AI will replace the people who don't know how to use AI. And it is time to adopt, and it's a time to adopt and learn this AI. And these kind of programs are a need of the hour. In fact, we have realized this, like, okay, and we are also establishing the Center for AI, ML, Drug Discovery, and Computational Biology in many institutes. So today, AM is a game changer. And whatever I explain, I'll be giving a talk tomorrow. So thanks a lot for giving us an opportunity to collaborate with you, and thanks a lot for this opportunity. So let us see tomorrow, and we'll talk in detail about how the AML is changing on every technology in detail. Thank you.
Thank you so much, sir, for your encouraging words. With that inspiring beginning, we officially inaugurate the Faculty Development Program on Revolutionizing Healthcare, the role of AI in drug development, clinical trials, and practice. On the behalf of organizing committee, I extend heartfelt gratitude to AICT Atal Academy, Molecular Solution for their support, our director, dignitaries for their encouragement, and our coordinators, faculty members, and all the participants for joining us in the academic endeavor. May this FDP inspire innovative ideas, foster fruitful collaborations, and drive meaningful research at the intersection of AI and healthcare. Now, I would like to hand over the session to Dr. Nidhi Mishra ma'am to take the proceeding forward.
A very good evening to all of you. And now we will be having the first talk of this program. We have Dr. Muhammad Imran Siddiqui ji with us as a keynote speaker. He is a chief scientist and professor in Academy CSIR. He is in charge of Center for Bioinformatics and Computational Biology at CSIR CDRI Lucknow. Dr. Siddiqui has been associated with CSIR as a scientist since 2004 and is currently working as chief scientist. He did his graduation in human biology from AIMS New Delhi and his master's and PhD in biophysics from AIMS. He carried out his postdoctoral research in the field of computational biology and chemistry at University of Würzburg, Germany and National Research Council Biotechnology Research Institute, Montreal, Canada. His research interests are bioinformatics, chemoinformatics, and artificial intelligence, machine learning driven rational drug design. Dr. Siddiqui has notable awards and honors to his credit, including BMBF Fellowship in 2000-2003 by Government of Germany, NSERC Fellowship by Government of Canada in 2003-2004, the Scientific and Technological Research Council of Turkey Visiting Fellowship in 2018 and very recently in 2023. He has delivered more than 75 invited talks in national and international conferences and workshops, including prestigious ICTP UNESCO conferences on drug design and development. He has been part of the conference held at Italy ICS UNIDO on drug development for developing countries. Dr. Siddiqui has been a constant visitor to CSIR South Africa. He has been a visiting consultant at ICS Unido Trieste, Italy. He is a visiting scientist at Ankara University, Turkey, where he has made visits in 2018 and 2023. Dr. Siddiqui himself has been organizing a number of training programs and workshops in the area of bioinformatics and computer-aided drug design and has supervised about 20 PhD students for their doctoral research. Dr. Siddiqui has, to his credit, more than 200 research papers in reputed international journals, and Dr. Siddiqui has been a continuous source of encouragement for bioinformaticians in this part of the world, that is the northern India. He is one of the key workers in the field of bioinformatics. We welcome you, sir, and we would like you to start with your talk, which would be very much informative and encouraging and maybe an eye opener to the beginners.
Thank you so much, Dr. Nidhi. It is really it's a wonderful opportunity to be here again in a program that is organized by your institute, and of course, this is highly useful, it's a very important topic that you have selected for this year's FTP. I must thank you for being part of this event. I can see my friend Raghu also in the audience. I must thank for supporting these type of events. It's he has been a good acquaintance for many years now, and I know his contribution to this development of this field in India. Good to see you, Raghu. So, so the topic of this FDP, I think this is very apt, and also it is very timely that we discuss how things are moving in the area of computational drug discovery, how it is transforming the classical computational chemistry to AI, and then now we are discussing things on quantum computing. So let me begin my formal presentation. I'm not going to be very, it is not going to be a technically uh, uh, complex presentation. I believe that many of the faculty participants from various organizations are not exactly in this field. So I'm not going to talk complex mathematical equations or something like that. It's going to be a simpler version of to try to familiarize our colleagues with what is happening in this field in more uh, general terms. So let me just try to. Okay, can you see something at your end?
Yes, sir. Yes, sir. Yes, sir. We are able to see.
Yes, sir.
You have to just
It's not
maximize.
in the maximize it. Yeah. Okay. So is it fully visible now?
Yes, sir.
Okay. Sorry about that. So now we know that in recent times, computational procedures are widely adopted in drug discovery and pharmaceutical industry, and most of you are well aware about this fact. Now we know that it's not in something which is it has become an indispensable tool in long process of drug discovery and development. It provides options for understanding chemical systems in different ways, yielding information that is not easy to obtain in laboratory analysis with considerably less cost and effort than experiment. So it is already a well-established scientific discipline. As we know, it is critical in the development of new treatments, has created enormous opportunities to speed up and rationalize the multidisciplinary drug discovery process, and provides us novel approaches to design and development of drugs. Well, the main objective of computational drug discovery is development and application of integrative AI, both machine learning as well as deep learning, also the bio and cheminformatic methods that can be translated to support drug discovery process by designing novel drugs, and with the ultimate aim of improving the human health. So, if we recall, go back in the recent past, not so distant, we used to do modeling using these something like what you see in on your screen is these plastic models. I'm sure many of you must have worked with these some toys with spherical attributes representing different atoms. These are colored by different atoms, and then there are cylindrical rods which represents the bonds between those atoms. And with these, you make some molecules by making combination of various atoms and different combination of different atoms, and by building you make a, well, these were actually the plastic models that we used to do modeling in earlier times, and these physical models were mostly based on molecular connectivity and about the stereochemistry of those molecules that you are working with. They were they gave some impression of the molecular dimension and also about the shape of the molecules that you were dealing with. However, they were not very accurate. They were difficult to manipulate and also store. So suppose you have a box of those physical attributes and you have to make a model out of that. You can use only limited number of models by using those plastic models. And you make a model, you have to disassemble it and then try to make another model using the same set of atoms and the bonds. And that way, so it is difficult to manipulate also. Suppose you make a complex model and you made a wrong selection of atoms. In place of nitrogen, you put a sulfur or some other atom. So you have to dismantle the whole model again and try to build it again from the beginning. Often fragile and static in the terms. You make a model and suddenly it falls on the floor, and then you have a problem that it breaks, or one atom goes in different direction, other in different direction, and so the model were sort of little fragile and not very robust. They gave very little information about the energy and other properties using looking at these models. So the chemical representation was really limited why if you use to build models using this
Sir, could
participant,
not see the presentation, sir. Someone is presenting and annotating. Sir, could you please share your presentation once again, sir?
your presentation is already there.
Ah, yes, sir. Yes, sir. Now it's visible. Thank you, sir.
So these models, they give very little information about the energy and also other properties that are associated with these models. So we see that the chemical representation was very much limited using these models and very difficult to construct large models. So these models were good to build small models like 4, 5, 6. Smaller models, but if you want to make a big want to build a larger models, then you have a problem by using these physical or mechanical ways of building models. Now, today we rely mostly on the computers. Computers allow us to do the molecular modeling. Modeling allows access to the molecular information, and these are useful for predicting properties and as well as how you use these properties for various application. So, whether a molecule has a chair or a board conformation, you can simply use the chemical programs and try to see what are the properties associated with this. You see how a protein is formed with a different set of chains, how it's what is the tertiary structure of this, like how a protein interacts with the DNA or RNA or many ways. You can simply use a computer using molecular modeling approaches and try to learn these things in a more simpler way and a simpler and less complex way as compared to the earlier versions when we used to do build models using other mechanical ways. So why do we need to build models in first place? Well, this is true for any field, not specifically for drug design. It is true for any field where the three-dimensional shape of molecules and their properties are important, can usually benefit from modeling. So, for example, the aircraft manufacturer, simply cannot go, they do not go directly into the workshop and start building their aircrafts. They do lots of modeling involving every dimensions, every aspects that is involved in creating an aircraft and building, bringing it into a reality. For example, what is going to be the dynamical properties, what is going to be the tailspin, what is going to be the elevation, wingspan, everything has to be calculated, predicted, modeled even before even a single screw is being made, put into place in a workshop. This is true for the car manufacturer also. They plan, design everything. They do all sort of modeling before actual product is born. And a more simpler example, a township. You go and try to find where is your flat and how does it look like. The town planner, the planner will show you the flat A with the sample ready flat. Will see how is the location, what is the orientation of kitchen, balcony, whatever. And so you give a good real presentation of what is going to happen when that is actually going to be implemented. So scientists use models to see how things work before they can make the final product, and this is safer and less expensive, as we assume that will definitely help you losing lots of money in failure of any project. So modeling actually involves computation and the graphics. Typically, a modeling exercise can have prediction and visualization of shape and the properties of molecules. Molecules could be macromolecules as well as small molecules. You can compare the shape, various properties, how a molecule A is different from molecule B with a simple change in a group. For example, a loss of hydroxyl group in a one molecule can alter the shape and the properties of the whole molecule, and uh, one could be a blockbuster drug and there could be a simple failure. And so you can compare how are these differences in the shape and properties can be evaluated. You can examine and predict the molecular interactions, the reactions between molecule A and a protein, molecule A and with nucleic acids, and so on. You can investigate unstable and excited molecules, modeling of dynamic systems, vibrations, diffusion, and one. So don't worry about this technical jargon. So simply consider that Basically, modeling aids in understanding the prediction of molecular phenomena underlying any given system. So that was the the computation. One of the important aspect of this modeling process is how do you visualize these models, and so that the computer graphics comes to your rescue. This allows you to visualize the models. You can easily alter, display, you can change atom, you can rotate bonds, color properties. So for example, in this panel you can see there is a guy in the red color. Now suppose this molecule, this residue is an important part which plays a role in binding of a, so this represents a animation of a binding site of a protein. And tyrosine is one of the important aspects. Suppose tyrosine is an important residue in this binding site. And if you mutate this tyrosine to phenylalanine, and that tyrosine is supposed to be important for binding to to a drug or drug-like molecule, the loss of hydroxyl group will make the, the molecule ineffective. So you can simply click change mutate tyrosine to phenylalanine and see how it is going to affect the binding. So you can easily alter and display, change atoms, rotate bonds, color properties. And so the spatial representation has tremendously improved as compared to the previously used models, like I mentioned in the physical or the mechanical models. And in a way, they are really highly interactive. You can simply sit in term of your molecule and you can command that I want to change it to and I want to change and mutate it to some other residue and try to see effect how it is have on the overall complex. So the chemical properties represented mathematically by evaluating equations in a computer program, AI machine learning models have been achieved, and good chemical representation achieved as compared to the previous ways of modeling. Now we go back slightly back around 10, 12 years back. If you recall, there was a Nobel Prize in Chemistry awarded to three these gentlemen, Michael Levitt, Martin Karplus, and Ariel Warshel. And the headlines of the newspaper was that three get Nobel for taking chemistry to the cyberspace. And what does that mean? That means that chemical, if you visualize a chemical experiment, you will visualize a chemist holding test tube in one hand and another a test tube but flask in another hand and mixing reactions, and you will see certain fumes coming out of those reactions. So this is what how we will visualize chemistry. So the chemical experiments ideally take place in labs, but the ability to carry them out in real time through computers, they have won Lewitt, Karplus, and Warshel this Nobel Prize for Chemistry in 2013. So today, the computer is just as important a tool for chemists as the test tube. That was the Royal Swedish Academy of Sciences that said in 2030 while awarding these three gentlemen this prize. Their research in 70s help us to understand how exhaust fumes are purified or how photosynthesis takes place in tea leaves and so on. This kind of knowledge makes it possible to find the best design things like new drugs, solar cells, catalytic converter, and many, many other applications. So, chemicals earlier used to create models of molecules using plastic balls and sticks, which is now done in computers. And the award of these three gentlemen in 2003 highlighted these aspects and acknowledged why the computers are so important in today's era in this field. Also in 1972, there was another Nobel laureate, American biochemist Christian Anfinsen. And many of you know that in his 1970 Nobel Prize acceptance speech, he said, I quote, one day it would be possible, he said, to predict the three-dimensional structure of any protein merely from its sequence of amino acid building blocks. With hundreds and thousands of proteins in the human body alone, such an advance would have vast applications, offering insights into basic biology and revealing promising new drugs. And so, what he prophesied in 1972, this was materialized in July 2021. While attempts to develop computer-aided models capable of solving the protein folding problems have been ongoing for many decades, but the accurate protein prediction has actually eluded researchers until these two seminal papers published nearly simultaneously in Nature and Science in July 2021, which represented the AI-driven software program called AlphaFold 2 and Rosetta. And I'm sure all of you now know what are these. Respectively, they can churn out accurate protein structures by thousands and advance that realizes and Stream 50 years later. So what this gentleman prophesied in 72, it came into reality thanks to the AI-driven software programs and all those advances that have taken place over the past those many decades. And so, after 10 years or 11 years, those three gentlemen got the Nobel Prize in chemistry. Again, in 2024, this Nobel Prize in chemistry again went to these three gentlemen, David Baker, Demis Hassabis, and John Jumper. And this year's prize was about proteins, that is, life's ingenious chemical tools. They cracked the code for proteins' amazing structures. David Baker has succeeded with an almost impossible feat of building entirely new kind of proteins. And both have, Hassabis and Jumper, they developed an AI model to solve a 50-year problem predicting proteins' complex structures. And these discoveries hold enormous potential as we see and also discuss in later slides as we progress further. So why do we study the protein structures? What is so special about the three-dimensional structures of a protein? Well, the structure of a protein is directly related to the protein's functionality, probably when determining it. And so the reasons for research in 3D structures are one, the most foremost in medicine, and the reason why we are holding this faculty development program. In the medicine, we can understand how biomolecules function and also interact, and underpins efforts to develop new drugs, in medical treatments, and understand to treat diseases. We can find targets for docking drugs, and these are one of the important aspects. I think you will hear more on these during subsequent lectures from various other experts in this FDP. And also there are other applications in agriculture or industry, which we are actually not uh, particularly bothered about at this stage, and we'll focus mostly on the first application. Now, you must be wondering, how do you get these three-dimensional structures? Well, there are three biophysical methods which helps you in studying or getting the structure of these proteins. One, many of you might be well aware of this X-ray crystallography method, and this is an important biophysical method where you get a three-dimensional structure of a target macromolecules with proteins and nucleic acids, how a ligand binds to a macromolecules, what is the ligand binding orientation, and so on. So you crystallize a protein and diffracts an X-ray, and pattern is obtained, and this is translated into electron density map, and this is fitted, and then you get an atomic model in the coordinate system, which translates into a three-dimensional model that you visualize in a graphical through a graphical interfaces. Then there is a nuclear magnetic resonance spectroscopy. This is used for determining structure and functional groups of ligand, which are likely to be important for binding. Here you use the solution structures of a protein, nucleic acids, as compared to the crystallized structure which you use in X-ray crystallography. Besides these two, there is a cryo-electron microscopy method also, which is used for solving structures of transmembrane proteins, where it is difficult to get the the solution or a crystallized component. So you use the cryo methods to look at the three-dimensional properties and structures of those. Based on these these three biophysical methods and the data that you generate about the structures, you also do a molecular modeling or homology modeling based, where you predict the theoretical prediction of three-dimensional structure of a protein. So, so the alpha fold is one example, or Rosetta, and then of course you have a classical models, a modeler, or various other programs that help you in studying these three-dimensional features of a protein. Of course, AlphaFold is worth mentioning here. This is an AI system which is developed by Google DeepMind and predicts the protein three-dimensional structure from its amino acid sequence. And it provides a high level of accuracy, which is mostly competitive with the experimental predictions. And one interesting fact is that in Cas14, AlphaFold was the top ranked protein structure prediction methods by large margin and produced prediction with very greater accuracy in terms of the prediction with very high accuracy. And of course, there is a Rosetta software which has a very numerous application in de novo protein design or enzyme design, ligand docking, macromolecular complex studies, and so on. So now, if we have been discussing proteins, uh, why? Because this is a part of what we call as a protein structure-based drug design cycle. Now, in this protein-centric structure-based drug design cycle, what you do when you start a drug discovery project, you look at this disease, what you are working on, you look at all the genetics, the physiological, every aspect of disease, and then based on various pathways, the role of proteins, you make a choice of a protein target. Once you make a choice of a protein target, it's about its utility and its importance in any disease process. To try to find about the structure of that protein, you use crystallography or NMR or homology modeling and various, including AI methods. Once you have a protein structure, you can use screening of compound database against that protein target and try to find if there are certain molecules that fit complementary. They show some complementarily territory to binding to the protein active or binding site. And then in the way you score them on the basis of some scoring function in the binding site and then rank and try to test them in your experimental setup. On the other way, you look at the binding site of a molecule and try to use some sort of the features that represents the binding site. You try to design something completely de novo without any information about what kind of molecules could be made here. So de novo means that you are building something from the scratch. Here you use the generative AI approaches also to build your molecular scaffolds. You use different fragments, try to link them together and make a composite molecules, and then try to synthesize this if it's synthetically feasible, and then test in the system. Once you reach at this testing stage, you try to see if it is showing some activity. You look, designate them as lead compounds. Look at the structure of the protein ligand complex. Try to see how these molecules can be further improved in terms to improve affinity, specificity, other pharmacodynamics properties, toxicity. So this goes in sort of iterative way in number of cycles until you find something promising in testing system, and then it further progress into the later stages in drug discovery cycle. So how do you get these 3D structures? Well, there is a data bank. Many of you know that it is designated as a protein data bank, and this can be reached through rcsp.org/pdb, or you can also find links through Antres or Blast or some other links which can lead you to this database. This is a repository of protein three-dimensional structures, more than 200,000 protein structure files from various sources, from various organisms, from humans, from disease causing, pathogenic, non pathogenic, every source solved through either Excel crystallography or NMR. They mostly find their way into this protein data bank. So suppose we work on a protein structure at CDRI or NMR structure in CDRI in any pathogenic or human proteins, it finally the coordinates of those protein generally gets deposited in 3 dimensions in this protein data bank. So the structures from the coordinates from this data bank can be downloaded and then can be visualized and manipulated with various imaging programs like KIME, Chimera, Rasmor, and so on. There are a number of programs that can be used for playing with these molecules nowadays. So if you look at the growth of PDB over a period of years, then see from late 1970s, it has grown enormously. And currently, there are more than 200,000 protein structure files, and so that shows that the structural data that is available for us is so important that helps in progressing the overall drives the whole drug discovery process in structure-based manner. Also, it is important to know here that function of many proteins is to bind some target molecules or a set of target molecules and then perform these some functions. And some of you from medicinal chemistry may agree that one of the basic tenets of pharmaceutical chemistry is that the biological activity of a molecule is dependent on the three-dimensional placement of specific functional groups. And we call them as a pharmacophore. So in a molecule, the whole molecule may not be important, but there are certain atoms, groups, pharmacophoric groups that are important and helps in providing the whole molecule their activity. And by understanding these protein ligand interactions, these are mediated by these pharmacophores. Also, we also know that with a very few exceptions, the drugs interact with macromolecular targets, and these are most often with the specific binding sites of membrane bound or nuclear receptors, enzymes, transporters, or ion channels. So, mostly the drugs have to interact with one of these entities here. Now, if you look at this relationship between the drugs and the macromolecular targets, you can think of this as a relationship between a lock and a key. Here, the lock is your target, the key is your drug. So most of the drugs snugly fit into the target. So once you know the three-dimensional structure of a protein, and if you know the binding site of your your protein, that gives you the information about how information about the keyhole where you have to fit your key. So like you fit a key into a keyhole space, you fit a molecule, a inhibitor or a drug into the space which have a specific residues oriented or presented in such a way that it will make specific interaction with the molecule there. So the like a key that fits in the keyhole, the molecule also snugly fits to the target and then have their action. So this PDB gives access to the structures of some locks, but not all. So if you go to the PDB, you find which lock you are going to work for your protein drug discovery project. And mostly you can look at the PDB search and try to see if the protein of your interest, the structures of that protein are there in that. You can analyze this protein structure, and analysis will provide the details of the log. By understanding the log structure, suitable keys can be designed and identified using the computer programs. And this in layman terms or common terms we call as structure-based drug design. So you have a structure, the log structure from X-ray NMR or from modeling. You use computer softwares, algorithms, generative AI, and so other methods, and then try to find something which is a best binder to this, and that can act as a drug or as a key to your log by this approach. So this is what we call as a rational drug design, where the three-dimensional molecular structure is one of the important aspects and forms the foundation of this drug design approach. And this relies on the knowledge of you can classify them as a receptor-based approach or a ligand-based approach based on the knowledge of the structure of the target protein. And once you have, if you know If you rely on the knowledge of the structure target protein, you can classify them as a receptor-based approach, and you can exploit these receptor then for molecular docking, virtual screening, de novo design, and many other applications. And suppose you have only the ligand information available from you, the activity data about the ligand, you can use these ligand-based approaches for using Pharma for modeling 3D QSAR, AI, machine learning-based methods, and so on. So we'll see some of these in more details in later slides. So, the idea of structure-based drug design is to identify a better key for a given law. And this you can do by using what we call as virtual screening. Virtual screening means that you are doing something in silico. This involves computational filtering or a docking of a large body of molecules, which is a collection of a database. So you have a various scaffolds, different diverse collection of molecules or binders, and you have a binding site the characterized by various features in the protein. So the idea is to segregate binders from non-binders and then get hands on to those which have a best binding and best fit into the binding site. So if you can see in this example, in one of very earlier PhD project in 2002 by Priyanka, so she used Plasmodium falciparum dihydroorotate dehydrogenase. This is a protein from Plasmodium falciparum. So the active site of the protein you see here is the three-dimensional structure of a protein. The active site is characterized by these residues that you are seeing popping up here. And once you characterize the binding site, you blast a series of molecules into the active site. And so you see here the molecules getting filtered into the active site, and these actually are getting scored by some sort of scoring function, and these scoring function rank these molecules on the basis of their affinity and scores. And so you pick those molecules which are better bind, which are predicted to have a better binding affinity in terms of scoring to the protein, and try to pick those few binders and take them into the experimental assay. So here idea is to identify those binders that have a very high probability of activity in the biological system. So instead of simply assaying thousands and thousands of molecules, wasting money and resources, you make a rational effort to select only a few of these which have a higher probability of showing activity in a biological assay. Now, one way of understanding these interaction with the protein and light again is what many of you are definitely familiar with this term called molecular docking. Now, this phenomena in this uses explores the interaction that occur when a ligand could be defined as any atom, a ion, or a molecule which attaches itself to the docking site of another molecule, which usually is a larger protein, which is known as a receptor. So again, here I am going to bring another analogy, much like the space shuttle that docks with the space station. So you have a small molecule, a discover or some other space shuttle goes up into the sky. There's an international space center, or maybe Shubham Shukla goes up, taking a ride up into the into the space. There is a international space center already stationed there. There's a bigger partner. So you would imagine that this smaller molecule, it cannot simply go and sits anywhere on the space station. There is a designated docking site on the space center where this shuttle will go and binds in a particular orientation, and only then if it fits into that snugly into with the International Space Center, as you see here, there is an interaction between the sets of cosmonauts and astronauts and so on. Similarly, you have a receptor, it is a docking site, and the molecule to be a good drug, it has to, or any binder has to go and dock that particular pace in that particular fashion in a highly specific and specific and selective manner. So by understanding this interplay between the ligand and its receptor, These scientists are able to design drugs that would block the docking sites of the receptor and thus disable the functional properties of the protein. And ultimately, this technology would help us to cure some of the today's most devastating diseases. We'll see some examples as we move on. So mostly these docking programs uses the same principles to calculate bound structure. They look for the sites of interaction between ligand and the target. Besides, earlier we classified them on the basis of algorithm that we used to calculate these sites of interaction between ligand and target. So this fast shape mesh matching algorithm used by dog and UDOG and incremental construction algorithm, FlexX, hammerhead, SLY, and so on. Taboo search, genetic algorithm, which is mostly used in gold, Autodog, FlexiDog. Autodog is also one of the very popular tool that is being used by academic communities worldwide. Then you have Monte Carlo simulations, Affinity, MacDog, and so on. Distance geometry based talking. And so don't worry about these. These are just to give an idea that there are so many programs use different sort of algorithms, both classical, quantum, AI based approaches, and so on, and help you in understanding how a protein interacts with the ligand and a protein interacts with the ligand and whether you can study their properties and evaluate them as a drug or drug-like molecules. Now the protein is there for docking molecules. You should have some databases also. Now these databases also have progressed over the past 15 years, more than 15 years, from being a mere repository of the compounds to being a powerful research tool for discovering new leads compounds. And this, the growth of compound databases in drug discovery, also research fueled the growth in commercial software for chemical database management and publicly available compound repositories. I'm sure Dr. Raghu will give you more detailed flavors of all these things in his presentation tomorrow. Maybe I think I see in the timetable. Uh, uh, well, most of the sources of the molecules that we see here are actually derived from natural sources. So 70% of the drugs that are currently in use owe their origin to the natural sources. And these are from plants, herbs, bacteria, invertebrates, marine organisms, and so on. And then you have another pharmacophage like in libraries. These majority of the pharmaceutical companies have relatively large chemical libraries, which represents the cumulative synthetic efforts of medicinal chemistry within the company. For example, in CDR also we have our own pharmacophore-based library, which is developed by various medical chemists who have been working in this area for many decades and have a collective uh, synthetic database of various molecules. And also a newer aspect of chemical library compound libraries is the creation of libraries with maximum diversity, and so you have a diversity based like in libraries. To give you several example how you get molecules, for example, from natural plant based product based drug discovery for screening of natural product based libraries. There are numerous large collections of NPs available for virtual high throughput screening and development of AI/ML models. For example, is Coconut. This is a collection of open natural product. This is an aggregated data sets of the 700,000 elucidated and predicted natural products collected from open sources and web interface to browse. There is another catalog of NPs from Zinc database, AfroDB, Analyticom, ICC, Indofine, Impact, Nobel, TCM, Taiwan, and so on. It's a composed of over 80,000 entries, and which can be directly purchased from for your screening projects. And more importantly, there's a diversity screening libraries like which can be used for various screening projects like from ChemDiv, a set of drug-like compounds based on the concept of targeted diversity for hits finding screenings. Then you have Enamine, Real, and Made, Ottawa Chemicals, Thermo Fisher, Life Chemicals, Arsenic, Cambridge, Weybridge, and many, many more. So they have millions of compounds that are available in SDF format and can be easily screened through various tools against a designated targets in your project. Now, to give an example of how these methods are then used in real life in labs, for example, I give you an example from how a pharmacophore-based virtual screening can be used, was exploited to identify some human DNA ligase 1 inhibitors in my group a few years back. And this is an important enzyme with implication in cancer. This If you know a set of molecules and how they bind to a protein, there is a way you can develop a pharmacophore model based on which important the atoms these are actually play a role in binding to a protein. And so you translate those atoms into some sort of pharmacophore query, represent that this could be a donor atom, acceptor, and so on, and then define certain, you can restrain those groups by adding some distance constraint and so on. Once you make a pharmacophore model, you screen that against a library, here in this case, a Maybridge library, which is a collection of around 50,000 odd molecules. You screen them and try to find out which of these 50,000 molecules actually fit to these type of features. And once you shortlist them, you screen, look at what is the binding consistency of these with respect to the model, how these molecules are in the shortlisted molecules, how these are placed in relation to the protein and how they make interaction with the protein. So based on this, you do all those profiling, identify, select few certain top ranking hits for experimental assay, and then you identify some scaffolds which actually inhibits the protein, and then you identify those scaffold as the lead molecules and try to further go into iterative fashion that I mentioned in this protein structure based cycle. So the activity gives you the confidence that your molecule are actually not a theoretical molecule and they actually translate it into a activity in an experimental way. Similarly, in this check one protein targeted pharmacophore-based virtual screening, the structure of check1 was downloaded from PDB 3PA5 model is generated based on the binding features of a known molecule, screen a database, shortlisted molecules. So starting from 50,000, you come to say only 1,000 probable molecules, and from those 1,000 you further analyze and try to see which of them actually can be taken up into the experimental assay, and from there you pick up those experimental weights and try to optimize them in your further optimization cycle. Another example is how you identify PKNG. This is an serine threonine protein kinase, which is important for a crucial role in regulatory process within the mycobacterial cell and signaling cascade or infected host cell. And so, if you know the protein and how it binds to a set of molecules, so we have our own inhibitors designed in the chemistry department. Those molecules, they show some activity against the protein, and those pharmacophore features derived from this were used to identify a novel for new scaffolds, um, which mimics the features of the active molecules. And then you dog them, identify it, score them, and then find out which of them are actually going to be taken into in the experimental evaluation. And these ultimately lead to you some of the molecules which show good micromolar or some micromolar activity in the assay and are further optimized. Now, so far, this is mostly the computational, classical computational chemistry approaches. Now, with the advancement of AI/ML also methods, with the rapid explosion of genomics in chemical big data, we have seen AI-enabled machine learning and deep learning. They have become indispensable tool for drug designers to mine chemical information and from large compound databases to design drugs with important and precise biological properties. And they have certain advantage as compared to those physical models that rely on explicit physical equations like quantum chemistry or molecular dynamic simulation, machine learning. These approaches, they mostly use the pattern recognition algorithms to discern mathematical relationships between empirical observations of small molecules and then try to extrapolate them to predict chemical, biological, and physical properties of novel compounds with desired um, features and activities. So how this is integrated into identification of some inhibitors. Now here, for example, K nearest neighbor or naive Bayes random forest SVM. These are various machine learning classification models. So you start with, so for example, here is a het is histone acetyl transferase. This is an important epigenetic regulator with implication in cancer um, disease. Now, these are novel therapeutic strategy for treatment of cancer. So, with information about known inhibitors, you can design study for identification of new inhibitors using multiple. So, modeling models were based using multiple machine learning classification methods. Once these models are done, these are evaluated, and best models were used for screening databases. Consensus hits are identified, dogged, and you select best 10 hits based on this for biological evaluation and further take up into and look at how these binds to the protein and how these are evaluated in the experimental assay to further optimize them. This is another example of MTB in oil acyl carrier protein reductase. This is here again an integrated machine learning models have been used, and these models were then used for screening libraries, identifying molecules. Of course, it goes through a thorough computation analysis, even simulation studies, and before we select molecules for biological assays. And this ultimately leads to those molecules which are further optimized. And some of these molecules which have optimized have actually led to a more advanced stage of development in IND days, where we have predicted scaffolds and being filed as an investigational new drug and has been awarded mission projects to further work on this to improve the efficacy. More recently, the artificial neural network based modeling for identification of biological evaluation of base one. And this is a protein which is beta site amyloid beta precursor protein cleaving enzyme. It's a transmembrane aspartic protease, and this has shown as a potential is a possible therapeutic target for Alzheimer disease. Now, this is an aggravating disease, involves aberrant production of beta amyloid plaques in base one, which catalyzes the rate limiting step by cleaving the amyloid precursor proteins and generates neurotoxic amyloid beta protein that aggregates to form plaques leading to neurodegeneration. So here multiple ANN models were developed based on the available inhibitor information about the base inhibitors from multiple repository, including Campbell, PepChem, and the consensus prediction based on various models were applied, molecules were dogged, identified, and further picked the best molecules that are taken into in vitro evaluation to identify a low nanomolar inhibitors out of these. So this workflow utilizes a multi-stage virtual screening protocol for identifying potential base inhibitors by employing multiple AN-based models. Mostly the hyperparameter, these are tuned models, were assigned a task to virtually screen libraries and these a set of molecules consistently predicted and taken up further. And the best molecules were found to be in the IC50 range of 0.043 to 0.05, a really sub micromolar. So this shows how computational predictive models actually translate the virtual molecules into the real molecules when in terms of when it comes to the translational aspect of these approaches. Aldose reductase is also another important enzyme of all pathways and is responsible for aggravating diabetic neuropathy complication. Here, more advanced three-dimensional convolutional neural networks-based ALR2 inhibitors. These are classified by techniques using the snapshots of images captured from three-dimensional chemical structures with multiple rotation as input data. So Kushagra worked on developing these methods, and this was convolutional neural network-based architecture was trained on around 360 sets of image data with along each axis and further prediction on Mayberry library for each of these models. And subsequently, the biological evaluation and showed up five of these molecules as a promising AR2 inhibitors and and suggested new scaffolds for further optimization in this project. So if you look at the study of the success story of various structure-based drug discovery methods, then you will come across certain molecules like captopril, which was developed by targeting angiotensin converting enzyme for treatment of hypertension developed by squid, or HIV protease targeted lopinavir, eluviran, or nelfinavir viracept. These are antiviral developed by Abbott and Aguran in collaboration with Lilly. Then there is another blockbuster called zanamivir, marketed by name of Relenza. It's an influenza neuraminidase. It is an antiviral developed by Monash University in collaboration with Glaxo. Welcome. So these involve lots of computational inputs while developing these molecules through these pharmaceutical companies. And more recently, when the AI has started making more inroads into the drug discovery process, we have seen lots of rapid advancements, developments. For example, in early 2020, there is a company called Exscientia. They announced the first ever AI designed drug molecule as an immuno-oncology treatment for several types of tumors to enter human clinical trials. In February 22, in Silico Medicine, another AI-based company, they reported the start of phase 1 clinical trials for the first ever AI-discovered molecules for idiopathic pulmonary fibrosis based on an AI-discovered novel target, all done at a fraction of time and cost of traditional preclinical programs. Details can be found at insilico.com. Subs- Later on in January 2023, AppScience became the first entity to create and validate de novo antibodies in silico using generative AI approaches. In the same year, the FDA granted its first orphan drug designation to a drug discovered and designed using AI to in silico medicine for global phase 2 trial for the drug for idiopathic pulmonary fibrosis. And so you see that the list is already growing. And we have seen a host of AI tools are impacting nearly every stage of drug discovery process, offering huge potential to reshape drug discovery research with the promise of lower cost and shorter development timelines. We understand that AI-enabled drugs discovery offers massive potential to enhance the development of new drugs and to treat potential incurable conditions. So I end up with some of these interesting but harsh facts. You know that the traditional drug discovery is a highly laborious and costly process, with average cost to bring a new drug to the market is estimated to be about more than $800 million. Excuse me. Mostly, generally, a new drug generally takes up to 10 to 12 or 15 years to progress from discovery to launch, and over 90% of the drugs that entered the clinical trials fail to make it to the market. This is harsh, but a fact, harsh fact, but it's a reality. But over analysis of over past decade or so, it has uh, uh, have shown the impact of computational modeling on drug discovery. We have seen that all these methods speeds up the key steps in drug discovery process by combining aspects of structural bioinformatics, structural biology, structure-based design, and now AI/ML approaches. And so the application of all these modeling in drug discovery and development is expected to reduce the annual cost of developing a new drug by nearly 33%. If you reduce, and also the time taken for drug discovery by nearly 30. So if you reduce the time by 30%, that is from average 10 to 12% to say 6 to 7%, of course, that the money that goes into this also gets cut by almost similar. So the example of like HIV protease inhibitors, they have developed, came into market in nearly 6 to 7 years of research, and that highlights how the overall process of modern drug discovery is being reshaped over the past many years. So uh, I think I should end here. It's almost 1 hour now. I'm
Thank
happy to take questions
Thank you
from
so much,
audience,
sir,
please.
for your talk. Now, the audience, you can ask any of your queries to sir. He would be more than happy to answer those questions. Please, you can ask questions one by one.
I know I kept it a very highly paced presentation, but I think many of you will. There are subsequent speakers in this FDP. They will go delve into more details about some of these process which I have just touched upon and given a flavor of what exactly is the advances in the um, modern drug discovery driven by computational aspects approaches.
I once again ask the audience if they have any queries, they can either type it in the chat box or they can ask their questions.
Hello, sir.
Yes, Anuj.
Hello. Yes, sir. Sir, good evening. Sir, I am having one query that whether right now is there any drug that has been designed by using artificial intelligence and right now it's in pipeline?
Yeah, you see that in the last slide I gave you some examples of how well this is still a developing field, and I must say that it is very fast-paced development. Already,
Okay.
if you start on a drug discovery project in 2020, and by 2023, if you have something in already in phase one or drug or in close to market, I think that highlights the
Okay.
promise
Okay.
that is there in this these approaches.
Okay, sir. Okay. Okay. Yes. Okay. Thank you. Thank you very much. Thank you.
Hello, hello, hello. Yeah, please go ahead. Are you listening me? Yeah, yeah, please go ahead. Yes, I have a question to Dr. Siddiqui. Are you listening me? Yeah, yeah, please go ahead, Dr. Sugandhi. Yes, what role does PBK modeling play in predicting drug infecting DDIs in clinical settings, and how can ADMET properly inform this model? Dr. Rameshwar, I am sorry, I didn't clearly hear what you asked. Would you like to repeat? What role does PBPK modeling play in predicting drug interactions in clinical settings, and how can ADMET profiling inform these models? Oh, so you asked about pharmacodynamics and pharmacokinetic properties. Yes. Is it clear? Yes. Yeah, of course. I didn't touch upon this aspect, but I'm sure there will be other experts in the panel who will be discussing more on this. There are, of course, the advancement, the QSR, the QSTR, the QSPR. These methods have reached, of course, now we have seen use of highly predictive AI machine learning tools that are very important and predict the toxicity and admit profiling of a molecule, how the molecule drug or drug interacts with other drugs or drug interacts, what are the how different properties can be modulated and affected by calculating various parameters. And this is again, it's a complex subject, but thanks to various advancement in both hardware and software dealing with this area, we are now in a stage where uh, uh, we can have very confidence in the predictions. For example, there is a method, the software called LeadQuest, which help you in predicting the toxicity profile of a molecule or various ADMET properties that are associated with. And so these you can predict while in the course of molecular design. So simply you cannot, you don't have to wait until the end of the design process. You can start applying these filters right in the course of molecular design and find out which molecule is going to be toxic or not fitting into the ADMET profiling, and those can be dropped instead of going later stage of attrition at a later stage.
Can I add something here, sir?
Yeah, please, Raghu, please go ahead.
Yeah. So, like today, like okay, I think like in the recent months, like you might have seen a lot of FDA things. Like now they are telling no more animal experiments, right? Because the reason is like okay, 90% of the molecules, even if the molecule succeeds in the preclinical trial, that is in the animal trials, the moment it goes to the human trials, it fails. So because of the genomics things and everything. Now, even in the since you're asking for the pharmacokinetics and these things, so what is the importance coming in here is like okay, even the population kinetics has to be calculated. Today, the technology is developed. Second thing is like in the earlier things like all the admin tools were predicted based upon the animal data. Today it is like okay, there are companies who has partnered with so many pharma companies and they are bringing the clinical trial data and nearly 220 parameters you can calculate. And the other beauty here with the AML coming in is now like when you're calculating these 220 parameters in any one of the parameter, like let us imagine toxicity or the absorption or any of those things is not matching up to the criteria. The AML can suggest like, okay, what is a functional group you can modify and add it there. That's the way like the AML is coming and suggesting because it is learning from the learned models. Like, okay, if I want to optimize a particular property, what is a suitable functional group, which is again considering synthetic feasibility and everything. That's the way the AML is coming and playing a lot. Probably I'll talk more on that tomorrow, but like I'm just giving, since the question came, I'm just giving a glimpse of it. Yeah, thank you.
I'm sure Dr. Raghu have lots of tools to address, and also this will excite your curiosity about these topics. I think he has a plethora of tools that address various aspects of drug discovery, both driven by classical methods and also the integrative AI ML methods. So we have another any other questions from audience?
May I ask one more question?
Please go ahead.
Can you elaborate on how CDRI utilizes PPK
Sorry,
distribution to optimize
Ramesh, but
dosage? Can you elaborate on how CDRI utilizes PBPK distribution to optimize dosage regimes for special populations such as pediatrics or geriatric based on ADMET database, and what benefit this approach offers in the development?
So, continuing with your with the explanation Dr. Raghu provided, actually there are, of course, we also use our own methods indigenously developed within our. So these are actually all based on how good models that you prepare. So what you can at the end, it all depends on what is the data that you are going to use for building models. If you have a good data, of course, your model prediction definitely will going to be good and will be useful. And suppose you have a bad data, the models will not be so accurate, and so you may have a misleading. So We try to use as best data that is available to model and then use to build patterns and whatever information from there and then try to extrapolate and then try to use. Of course, CDRI, we definitely use these applications. I cannot give you particular examples, but almost every drug discovery project, these methods are used very much, and of course, these methods definitely help in fast tracking various projects.
Thank you. Thank you very much.
So audience, do you have any more questions?
Thank you very much.
I think we should conclude this session. Siddiqui sir, I would like to thank you on behalf of all the organizers, Triple IT Allahabad and Molecular Solutions Private Limited. And your talk has been very informative, and you have been a constant source of encouragement in this field since 2003 2004, and we have been benefiting from your talks since last so many years. Many thanks for accepting our invitation always, and giving us guidance in the form of lectures and all kind of suggestions. So I would like to thank you for being a keynote speaker in this AICTE sponsored Atal 6-day FDP on revolutionizing healthcare. So many thanks to you, sir. And
Thank you so much. Yeah, thank you so much, Dr. Nidhi, and I must thank all the organizers for bringing the experts and organizing this wonderful and highly useful event. I wish you all the success and happy participation for the participant for the rest of the sessions. Thank you so much.
Excuse me, sir. There is one question in the chat box. What is the
Yes.
major role generative and predictive modeling in computer assisted drug delivery, such revolutionizing in pharma industry?
Well, like the drug design, the delivery also is a very important aspect. The drug delivery, of course, here you have a different set of parameters to be modeled. So, for example, a molecule has to go through once it is in vivo, it has as compared to the in vitro behavior, it behaves differently in the walls. So once it is in body, it has to come across lots of different challenges. There are different barriers it has to cross. It has to be targeted to a particular area. Now, all these drug delivery aspects also are these are models. There are different models that parameterize and models. These are built and then these are fitted into see how a molecule could be uh, could be targeted to a specific target and what are whether it can be delivered, it has to be transported in such a way how it can be transported. All these properties can be predicted beforehand and evaluated. Of course, this also can be done while in the course of molecular design. All these properties are evaluated.
If you want, I can add two more.
Yeah, you can please go ahead.
Yeah. So generative AI and predictive AI. So generative AI plays a very crucial role, particularly when you're designing any molecule for your target. So, for example, like okay, you can give a target structure. This is my target structure. Can you design something? Okay, you can ask the generative AI because the generative AI has been trained based upon multiple billions of structures and other things. So what it does is it looks into it and point by point on a fragment by fragment it generates. Or like, okay, you can say like, okay, this is my molecule. I wanted to change only an R group. Okay, you suggest me what is the best R group. Or you can say like, these are my R groups. I want to change only the scaffold. So generative AI, what it does is it goes and searches from the databases of scaffold, because when it's coming and replacing a scaffold, it should match like all those R groups, everything. So that's where the generative AI comes into play. And not only in that, like it also helps in the synthesis part of it, like particularly in the process chemistry, in the retrosynthesis and everything. Now there is a Bayesian optimization comes in, where the generative AI helps in like, okay, at each and every step, where I can optimize to find a better route or a cost-effective route, or I can bring in a much economical synthesis or anything. That's where the generative AI comes. The predictive AI comes into more into the AID, as Dr. Siddiqui was telling, admit part of it. So where like, okay, you predict, like, okay, you have trained the models based upon various methods where Dr. Siddiqui told, and like, okay, given your molecule and how you predict it. So all these things, again, when it comes to AI, it all depends upon the data, the accuracy of the data, the source of the data, how curated this data, everything. It depends upon it. We can talk more about that tomorrow. Like I'll be talking still more on these aspects.
Thank you, Dr. Raghu.
Thank you, sir.
I think there are no more questions.
Yes, sir. This session is about means almost ended, and I would like to thank you again. And the next speaker would join at 8 p.m. So we have almost 13 minutes. The participants can take a break.
Okay. Okay. Bye, Raghu, Dr. Nidhi.
Okay.
Excellent session, sir. Thank you.